Good morning, good afternoon, and good evening. Welcome back to Chapter 18, the animals. Let's talk about our first group of animals, and let me keep a running total here for you. So remember, what domain are we in? We're in domain Eukarya. How many kingdoms are in domain Eukarya? 4. Plants, animals, fungi, and protists. We've talked about protists, we talked about plants, we talked about fungi. So what kingdom are we talking about? We're talking about kingdom Animalia. All right? Now we're going to talk about the different phyla within kingdom Animalia. And the different phyla, our first phylum is phylum Porifera. Our first phylum is phylum Porifera. Poriferans are sponges. What does pore mean? What are your pores? Your pores are holes in your skin. So a pore is a hole. Fair comes from the same root word as like ferry, as in like a ferry boat. Fair means to bear or to possess. So poriferan means hole bearer. It actually means little hole bearer. But it means hole bearer. They have holes in them. Sponges do. There's about 5,000 species of sponges. Remember, they have that asymmetrical body plan. They have no body plan. They're mostly sessile filter feeders. What does sessile mean? Sessile means they don't move. They don't move. They are a filter feeder. What does filter feeding mean? Well, filter feeding is the simplest form of nutrition, the oldest form of animal nutrition, and that is that they just simply draw in the water around them. And then in that water, there's going to be floating particulate matter, floating stuff, bits and pieces of fish that have been eaten by larger fish. There's going to be poop. There's going to be tiny little bacteria. There's going to be tiny little protists. There's going to be all kinds of organic matter floating around in that water. A filter feeder pulls that water in, sucks that water in, and then, as the name implies, filters it. They send that water through a filter. Anything that's a particulate matter is going to get caught on the filter, and then the now clean water gets pushed back out. And they eat whatever the filter caught. Whatever they caught on their filter, that's what they eat. And I'll show you how it actually works as sponges in just a little bit. But there are lots of groups of animals that are filter feeders. Lots of them. Why? Because it's easy. You don't have to hunt anything. Food literally comes to you. And remember, most living things live in the water. So there's lots and lots of water. There's plenty of space to be able to filter this, to filter out food. Problem is there's not a lot of food in the water because there's a whole bunch of filter feeders. So it's very cheap, it's very easy, you just don't get a lot of food. And so most filter feeders tend to be small. It actually takes sponges. They've got to filter 700 or 70 gallons, 280 liters to be able to grow a single ounce, to put on 1 ounce. And an ounce is about how big that dipping cup is that you get of ranch dressing from McDonald's. That's about 1 ounce. Okay? So to put on that much weight, a sponge has to filter 70 gallons of water to be able to grow a single ounce. They can be in size from a few millimeters all the way up to 2 meters tall. Big sponges. Big sponges. Sponges don't have any true tissues. So all they have really are just 3 different cell types. First cell type are called choanocytes. Cyte means cell. Choan means collar. So choanocytes are collar cells. They've got a collar. I'll show you what the collar is in just a little bit. They have a collar, and within this collar, there's a flagellum. The collar kind of looks like the cone of shame from Up. Remember when Doug the dog has the cone, right? Cone of shame. That's what a collar looks like. Inside that collar, there's a single flagellum coming out, a whip. And that flagellum creates current. When you think of flagellum, think of like a sperm cell. And the sperm cell, when the flagellum spins, it pushes the sperm cell through the environment. All right. Well, the collar cell is actually embedded within the tissue or within the body of the sponge. So when the flagellum spins, the cell can't go anywhere. So if the cell can't go anywhere, that means the water has to. It's kind of like a boat that's up against a dock and he revs the motor. What's going to happen? It's going to shoot out a whole bunch of water behind it because the boat can't move, so the water has to. When the chelanocyte spins its flagellum, it moves the water. The water is drawn into the sponge. Anything floating in the water, the particulate matter, gets caught on the collar of the sponge, of the collar cell, and then the clean water gets pushed out. Our second kind of cell that we find in sponges is called an amoebocyte. The job of the amoebocyte is to go to the collar cell, the chelanocyte, and to get food from it. And then to take it to our third cell type, which is called an epidermal cell, a skin cell. That's basically all of the cells that we find in a sponge. That is basically how complicated a sponge is. I've got a cell that gathers food. I have a cell that's a waiter that picks up food from the collar cells and takes it to the skin cells. Skin cells are what hold the animal together. Amoebocytes go back and forth, getting food from the collar cells and feeding the skin cells. And that's it. That's how complicated a sponge is. So this shows a simple sponge. Right here we can see overall how the sponge is made. If it's a sponge, it's got holes in it. So we see the holes all over. Those holes go all the way through. This right here is an extreme blow-up of what the tissue, what the body of the sponge looks like. Here's that collar cell. See the collar right here? There's the collar. Coming out from the center of that collar cell, so just basically imagine a lampshade, and the stem of the lamp is the flagellum. The flagellum right here comes out from the center of the collar cells, and it spins. And when it spins, it creates current, and this draws water from the outside. These are those epidermal cells. The skin cells, the outer skin cells. It draws current from the outside. The water comes in, passes past these collar cells, and the collar cells grab onto particulate matter. Any bits and pieces of anything are going to stick to the collar cells, and the collar cells then eat that. The amoebocyte will pick up food from the collar cell and then deliver it to the epidermal cells. And that's it. That's all a sponge is. We do have these skeletal pieces called spicules that are made by the amoebocytes. And it depends on what kind of sponge it is as to what those spicules are made of. But this is it. This is how complicated a sponge is. This is the simplest version of sponge. There are more complicated versions of sponges, but it's just variations on this theme. It's just a vase with holes in it. It draws water in, cleans that water, and then pushes the fresh water out the top. All these collar cells all work together at the same time, drawing water in. The water comes to the center, and then it's pushed out the top. And that's the life of a sponge. That is the life cycle of a sponge. Sponges are so simple that if you take a sponge and put it in a blender, and then pour the pieces out, they'll reassemble back into a simple sponge again. That's it, man. Sponges.